So we're going to move on to our next talk that's going to be given by Professor Florian Deisenhammer. Florian is a professor of neurology at the University of Innsbruck in Austria, and we're going to be continuing the theme of MS. And Florian's very much an expert on interferon-beta, so we're very much looking forward to hearing his story relating to that and the issues of immunogenicity. He's also involved in an EU project regarding immunogenicity in the context of the pharmaceutical industry as well as many academic groups. And so it's great to have him here representing Avarisk as well. We'll probably hear a little bit about that as well. So I will, without any further ado, hopefully get this to start. And I'll pass over to you.
Okay, thank you very much. All right, good morning. Good morning. Can you hear me? Yeah, hopefully. Thanks for the kind invitation to this very lovely town here. It's fascinating to be here. It's MIT just around the corner, and one of the major biotech companies this way. I believe it's Biogen, IDEC, and yesterday I've seen Genzyme is here, so it's really in the middle of the hot topic we are discussing here today. I have to apologize up front. The topic of interferon antibodies and immunogenicity interferon is very complex. I've been trying to understand it for almost 20 years now, and I'm not there. And this is reflected by the presentation. It's slightly chaotic, and every time it's not the first time that I'm giving this presentation here on interferon-beta antibodies, and every time I try to prepare it. I go back to the last presentation. I think, just take the last presentation and give it again. And then you go through the slides, and you can't do that. It's totally mixed up here. So I reshuffle this presentation every time I do it from the beginning. So this time I hope I'll be there, but I'm not quite sure. Right. So this interferon-beta, and this is some basic principles, of course, on drug immunogenicity. We have in the middle we have the immunogenicity issues, and then we have a lot of factors that contribute to the immunogenicity of a drug. You can see here its protein sequence has been discussed just in the previous presentation. Clumping is certain issues, contaminates during the production of the drug, the dose frequency, then of course patients are a very important factor in immunogenicity. It's genetically triggered, actually, and controlled how the patient reacts to the drug. And then if you give it into the muscle, or if you give it into the skin or somewhere else. That's all factors that we're going to discuss. But first of all, let's focus on the immunogenicity side of things. And the first question is, how do you measure immunogenicity? And we usually do that at least in clinical terms. In the clinical situation, we measure antibodies against the drug. So we have a drug and we have the antibodies, and by definition, semantically, at least in my understanding, this is an anti-drug antibody. So do you measure all these antibodies? And there's a relationship between different things you can measure in the drug response, in the immunogenicity response. And one thing here is that's the anti-drug antibodies. Often they are called binding antibodies because you just use an assay, you bind the antibodies to the drug. But it should be the same as anti-drug antibodies, so no difference here. So it's an interchangeable term, essentially. And there's a phenomenon which is called neutralizing antibodies. And of course, this is all related to the drug effectiveness. You can measure that, of course, in terms of pharmacokinetics. You can measure the drug directly, or you can measure the effects of the drug, which is pharmacodynamics. In interferon-beta, you usually do the pharmacodynamics because it's essentially impossible to measure interferon-beta in the circulation. So you lie to that. In this presentation, I will say almost nothing about this part of the story. I will focus on the binding antibodies and the interaction, the neutralizing antibodies, and what may explain this interaction between those two entities. if it's two entities, I don't know. So let's go back to the drug and the antibody situation. So the question is, if you measure drug antibodies and if the drug is actually affected by the antibodies, or the efficacy of the drug is affected by the antibodies, the question is it just a matter of the quantity of antibodies? The more antibodies you have, the more drug is neutralized, and then you have enough antibodies to immobilize the drug, and the drug cannot bind to its target. In case of interferon, it's the receptor. That's one scenario. The other scenario is, are there black sheep in the antibodies? So is it a qualitative thing? Do we have some antibodies, the red ones are just the binding antibodies, but they don't affect the drug at all. And there's a few others, the black ones, that are responsible for neutralization of the drug. So we can bind to the drug, doesn't affect, and then if there's the black sheep coming on, the drug is neutralized. And we actually don't know exactly if we can differentiate those two situations. But I'll give you some thoughts and ideas how we try to approach this issue. So the question, of course, is neutralization a quantitative or a qualitative phenomenon, or is it both? So is all PARPs neutralizing antibodies, just depending on the dose of the antibodies? Is PARPs totally different to neutralizing antibodies? Or is NAbs a subpopulation of binding antibodies? First thing you have to consider when we talk about neutralizing antibodies, we are not talking about neutralizing antibody. And this is because we can't measure neutralizing antibodies. This is how we do it. So this is how you measure interferon activity and how you measure antibodies in that situation. So interferon-beta, that's the blue symbols here, should be the interferon-beta molecule. And it binds to the receptor. The receptor is the yellow thing here on the cell. So that should be a cell here, and that's the nucleus. So we have the receptor on the surface of the cell, and interferon-beta binds to the receptor, and this is how it works. So what we can do now when we measure antibodies and neutralization of antibodies, we can do only one thing at once. So we can either measure the antibodies, which is on the left side, can be green antibodies or the red antibodies, but we don't know if these antibodies are interfering with the receptor binding of the interferon-beta molecules. We just can measure antibodies, that's the one thing. On the other side, we can measure the activity of interferon-beta. So what we do in the neutralizing assays is we add interferon-beta to cells, we measure the activity of interferon-beta, and then we add antibodies, and if the antibodies, and if there's a reduction of interferon-beta antibodies, interferon-beta activity, we assume that the antibodies are responsible for that neutralization, but we don't know exactly. So we can't really directly measure neutralizing antibodies. We always need the combination of antibodies and the bioassay measuring the activity of interferon-beta. So somehow the term neutralizing antibodies is misleading because we don't know formally exactly if it's neutralizing antibodies. We assume it from this experimental situation. Okay, so now let's try to explore somehow how this might be related, all this antibody stuff and the neutralization to each other. And this is some to start with. So in the last presentation, it was at the end of the presentation. Now it's in the beginning of the presentation. You see, it's very difficult to get a straight line into this. So we have what you should know is we have three different preparations of interferon-beta licensed in the treatment of multiple sclerosis. You've seen the disease in the last presentation. So we have interferon-beta 1b, which is not glycosylated. This is produced in E. coli, so no glycosylation there. And then we have interferon-beta 1a, two different preparations. One is applied by IM application, and the other one is applied by SC application, subcutaneously. Both are very, very similar, 1A, and they are quite similar to the human interferon-beta. So it's almost identical to human interferon-beta, apart from some side chains that are slightly different because it's produced in Chinese hamster ovaries. So what we have here is this is the number of patients here who produce binding antibodies. So there's immunogenicity. It's always difficult to talk about, you have to define immunogenicity. At which level do you want to measure immunogenicity? At which level is it relevant immunogenicity? And there's many different aspects, of course, coming in. If you just look at the immune response, you're interested in the binding antibodies. If you look at the relevance, clinical relevance of the immune response, you look at neutralizing antibodies. Is it really relevant for the patient? So what we see here is the response to the drug is different. We have quite high response here in interferon-beta 1b, which is interferon. And we have a low response to the 1a preparations, which is not very much different. When you look at the neutralizing antibodies, around 1/3 of those patients who develop binding antibodies against interferon have actually neutralizing or neutralization in the assay. And it's only 12% of all patients developing binding antibodies that neutralize against 1a IM, which is Avonex from Biogen just around the corner. And half of the patients who have binding antibodies being treated with Rebif, a subcutaneous form of 1a, interferon-beta 1a, develop actually neutralizing antibodies. So the potential of binding antibodies to neutralize differs between the different drugs. So it's one of the things we should consider. And then we looked, of course, at the subclasses. Is there anything in the immune response that might differ between binding and neutralizing antibodies? So that's quite early work. So we split the patients into patients who had neutralizing antibodies and those who had no neutralizing antibodies. So that's the non-neutralizing antibody group. And then, of course, as the antibody response changes over time, we tried to have equal time on treatment in both groups, so there's no confound in terms of the treatment duration. And what we found here is that the IgG4 response and also the IgG2 response was much higher, at least in terms of percentages, so higher frequency of these subclasses in the patients who had neutralizing antibodies compared to patients without neutralizing antibodies. And IgG4 is a subclass, an Ig subclass, which usually occurs in long-term immunization and promotes and provides long-term immunization, like in bee farmers. They get stung all the time by the bees, and the antibodies against bee venom is usually IgG4 after many, many years of repetitive stimulation of the immune system. So they switch totally to an IgG4 subtype. So this is quite similar here. Then also we looked at some kind of affinity, which is a relative affinity measure. I'm not going into detail here. And what happens over time, of course, and there's the time factor again coming in. Over time we have an increase of the affinity of antibodies in patients who have neutralizing antibodies. And there's a difference between Rebif 1a preparation, a slight difference in interferon. And I'll come back to this a little later. So we have a better affinity maturation with the 1a preparations compared to the 1b preparation over time. So there might be some selection going on for a certain subtype of binding antibodies, anti-interferon antibodies, that are then high affinity neutralizing antibodies. So there's some quality component to this whole antibody issue. And again, timing is very important because it depends very much at which time you look at the antibody response and how you want to relate binding antibodies to neutralizing antibodies. And again, this is early work we have done a couple of years ago. So we looked at patients who developed neutralizing antibodies and patients who did not develop neutralizing antibodies. So neutralizing antibodies is a relatively late phenomenon in this drug. It doesn't occur in the first month. It just occurs after 6 months at the earliest. After 9 months, after 12 months, you can actually measure high levels of neutralizing antibodies. Before 6 months, essentially, you can't detect neutralizing antibodies in the patient. So it's a very late development. And also that's something which is special in interferons. Anyway, so this is the binding titer here. This peak here is the binding titer in patients who develop neutralizing antibodies later on. This here is the neutralizing antibody titers in those patients. So it's down here. We have a boost in binding antibodies. It goes down to low levels, and then it parallels the titer at least parallels, but the neutralizing antibody titer, the binding titer and neutralizing antibody is close to each other. So depending on which time you measure neutralizing and binding antibodies in this patient, you would get totally different interactions and correlations between antibodies that neutralize and antibodies that do not neutralize. And this line down here that never goes really up is the binding titer in patients who do not develop neutralizing antibodies. So there's some quantitative stuff, and there's also some qualitative component to the selection of these antibodies that actually get neutralizing later on in the treatment. Also the binding site, of course, is there any peptide that is responsible for neutralization, and this is the idea. We have the molecule here, this is the antibodies. This is not really proportional because interferon-beta is much smaller than Ig molecule. Just a cartoon. And so the idea is there different binding sites that might induce different immune responses. And we looked into this again. So what we did is a very conservative approach. We chopped up the protein, the peptide, the sequence of interferon-beta. It's a 166 amino acid sequence. We broke it down into peptides of 12 amino acids. We started first at the N-terminal end, and then with an offset of 5 amino acids, we produced the next 12 peptides. So we ended up with 32 different peptides, and with quite some overlap between the different peptides. And then we looked at the antibody response within these different peptides in the patients with neutralizing antibodies and patients without neutralizing antibodies, just binding antibodies. So what came out here is 3 peptides that were of interest. There are 2 peptides that had that were against which we could find a higher frequency of antibodies, which is the very N-terminal end. So the first 12 peptides were binding antibodies much more frequently in patients with neutralizing antibodies as compared to patients without neutralizing antibodies. This is always binding antibodies. It's just that the stratification into the 2 groups is based on the neutralizing assay. But what we do here is we just measure binding antibodies against the peptides. So it doesn't tell you about the neutralization actually. And also on the very close to the C-terminal end, again, we found a higher frequency of binding in patients with neutralizing antibodies compared to non-neutralizing antibodies. And this is because this was the most frequently found peptide by the antibodies. So almost 2/3 of the patients identified, irrespective of the neutralization status, had binding to this peptide here. So interestingly, is the N and the C-terminal end, they stick out a little of the molecules, so they might be more prominent and more accessible for antibodies, and this might be one of the reasons why neutralization is promoted. And then we looked at the correlation between the neutralizing titer and the titers against these 3 peptides, and we found a high correlation between the neutralizing titer and the titer against the first 12 peptides, so the N-terminal end of the molecule. Highly significant, quite a strong correlation between neutralizing titers. So that might be really a responsible peptide for promoting neutralization against interferon. The other thing is, when you correlate between binding antibodies and neutralizing titers, you have to be very careful which assay you're using. So here we have the neutralizing titer, and here we have the binding titer, and this is 3 different assays we compared. These are homemade, just binding ELISAs, direct binding or capture ELISAs with an antibody stuck onto the plate, and then capture the interferon-beta captured, so it's sort of sandwich system, but also direct binding, so we just put interferon on the plate and measure the antibody response against this. So that was quite a nice correlation, not a big difference between these 2 assays with neutralizing titers. And this is a commercial assay we tested from Switzerland, which had a much less performance as compared to the homemade assays. So again, it depends on which format you're using for your assay when you want to look at the correlation between binding and neutralizing antibody titers. And also it depends, and this is very important, I believe, it depends very much on which titer region you look at. The correlation in low NAb titers, which is less than 100 neutralizing units, believe me, it's a low titer. I'm not going into detail how to measure or how to calculate the titer in neutralization, which is a poorly understood science made by a few people in the world. So again, but what we see in the low titer region, there's a very poor correlation between binding titers and neutralizing titers. Also in the intermediate range, which is 100 to 500 neutralizing units, which should be relevant for patients, at least transiently relevant for patients. Very poor correlation. Only in the high titer region of neutralizing antibodies we can see actually good correlation between binding and neutralizing titers. So again, that speaks for the selection of neutralizing antibodies out of the pool of binding antibodies, affinity selection perhaps, because you only find high titer neutralizing antibodies after a long time of immunization. So it takes a while to get high titer neutralizing antibodies. And again, if you compare the assays, you have to be aware of this phenomenon that the low titers is a lot of background activity in your assay that you detect. And this might relate to the fact that you don't find a clear correlation between neutralizing antibodies, low titer neutralizing antibodies, and clinical non-response or response against interferons. So it's this is really the relevant ones here. However, the binding antibody, as I showed you before, we have a peak early on and then it goes down and then it goes up again later. This is binding antibodies. There's a predictive value to that. If you measure that early, after 3 months, that's a recent study we've done in the previous EU project. So what you find here is after 3 months, it's quite reliable to predict later development of neutralizing antibodies. So in clinical terms, that might be quite a useful test to use to look at binding antibodies early during treatment after 3 months, which is very relevant because the loss of efficacy occurs at 2 years on treatment. So it takes a long time to have a clinical effect of the antibodies, because the neutralizing antibodies, because it takes so long for them to develop. So that's quite a good test early on to predict reliably a later development of neutralizing antibodies. I'm doing time-wise?
Yeah, next 5 minutes.
5 minutes. Okay, and that makes things even worse. There's neutralizing titers over time. We are looking at many, many years. That's 8 years. That's the median titer. And it's different between interferon-beta 1b and interferon 1a. So we have 2 different molecules with a different immunogenicity and a different course of development of antibodies. Okay, let's skip this one here. Now let's go away from immunogenicity and look at few other factors we have listed here and how what we learned from those. So that's going back to the molecule. This is interferon-beta 1a, so that's essentially the complete molecule with the glycosilation. 5 helices, 166 amino acids, and this is 1b. There's a lack of the sugar residues, and there's also 1 amino acid. It's short 1 amino acid, and 1 is substituted. So it's different, and of course we have different immunogenicity. So Avonex, this is the drug from Biogen Nidec, just back here. So they actually took care of this problem very early on. So they had an initial pivotal trial with their product, and they found quite a high frequency of neutralizing antibodies, more than 22%. So they went back to the lab. They changed the manufacturing. They removed the HSA, but be careful with the HSA. It turned out that the aggregation, oxidation was very much reduced after removal of the HSA, and they did some other things they are not telling us, but they reduced the immunogenicity to 2 to 5% in the following trials. And that's confirmed with many, many other labs and also in clinical routine. So they actually changed immunogenicity, changing the production process of the drug. And this is, to confirm all this, this is HPLC analysis, looking at HSA. This is interferon, so interferon-beta 1b, two different labels but the same drug. And also Rebif, at least in the US, they have a higher peak of HSA and no HSA in Avonex. Now, Rebif, the other interferon-beta 1a, they changed also their formulation. They removed the HSA, which is available in Europe. And they actually reduced immunogenicity. They didn't quite manage to reduce it to 2 to 5% like Avonex, but they reduced it by 5 to, let's say, 7%. So that's something to do with immunogenicity of interferon-beta, but be very careful. There's another example in drug history where HSA was removed, but that led to an increased number of antibodies. So that only applies, there's no, the problem here is that we might not have a general rule how to deal with immunogenicity across different drugs and across different populations, patient populations. So this is, I think that's really challenging. And this is one of the tasks we want to take on with the Aggregates project, to find common traits across different drugs and different patient populations. So we'll see what comes out of that. And that was quite dramatic because in erythropoietin you get pure red cell aplasia, which is a fatal disease actually. Frequency of application, this is from a off-label trial many years ago in Denmark. They looked at Rebif, which is usually applied 3 times a week, but they also looked at efficacy of once a week Rebif. What they found in terms of neutralizing antibodies, it's much less if you use it just once a week. So there's a dosing, of course, there's a dosing effect to this. But on the other hand, if you look at interferon in the different trials, they had an 8 million units trial, which is quite a high dose. They found 38% of patients neutralizing antibodies. A fifth of those, essentially the same frequency of neutralizing antibodies. So no real dose response here. Different populations, same drug, lower response in terms of neutralizing antibodies. So again, a bit of complex situation here, same drug, different populations that might influence the immunogenicity of the drug. There was a clear dose response in Avonex. So when they used 30 versus 60 micrograms, a clear increase of neutralizing antibodies. In Rebif, it was essentially the other way around. In the higher dose, they found a slightly lower, it was not significant, but they found a lower rate of neutralizing antibodies in the higher dose. So again, it's, as I told you in the beginning, it's a complex and very confusing situation. We don't really know what to make of all of this. But again, it shows us that we have to look at these different drugs in a different way when we look at immunogenicity. And just at the end, one of the pitfalls with biosimilars. So we were involved in a biosimilar study. We were asked to do the antibodies, and we did the antibody testing in this study. So this is Avonex actually, and this is the biosimilar. And we didn't find any relevant antibodies in this study. Not many patients though, but no antibodies in both. So they said, okay, low immunogenicity, new biosimilar, low immunogenicity, nice drug, isn't it? Then they looked at the bioactivity of the drug. And they couldn't find any bioactivity of the biosimilar. So no wonder it's not immunogenic because there's no drug in there. So you have to look at this. When you have look at this, you always have to look at all aspects of the drug, not only the immunogenicity side, but also on the efficacy side. That's what I want to tell you. That. So in summary, there's an association somehow between binding and neutralizing antibodies. There's some tighter correlation, but there's also some particular characteristics in terms of qualitative differences between NAbs and non-NAbs. Timing is always very important to look at. At which time do you do the tests? At which time on treatment do you compare all this stuff? Because it changes over time, and it's very important for interpreting of test results and reporting. So for us clinicians, We have to be very careful, and we have to know how long the patients are being treated, because that affects the reporting and consulting of the patients. You have a lot of different tests, but if you want to choose one single test in clinical routine, you choose the neutralizing antibody test, because it's the best. That's the best test actually in the clinical context, and to draw any consequences for the therapeutic decisions in patients. Why NAbs? Big question. Don't know really. It's probably breaking of tolerance. It's a human molecule. It's a cytokine. We have all in us, interferon-beta. It's slightly different. It comes in a very unusual dose. It's very, very high dose in physiological terms. And also it comes in a very, very strange way in your skin or in your muscles. So that's not the usual. So this is why there's probably a danger signal, and why immunogenicity, immune response is alerted. So we probably break tolerance over time. There's a huge difference between individuals. So there's clearly in individual disposition to develop antibodies against the drug. Of course, drug design, formulation, route of frequency, and application plays a big role in immunogenicity. That can be actually controlled. And measuring only one component in the whole issue of neutralizing antibodies, just one assay, is definitely not enough. If you want to get the whole picture, you have to do PK, PD, binding antibodies, neutralizing antibodies, to get an idea. Perhaps subtypes, affinity, all that stuff, to get an idea what it's all about. And this is the Aggregates project. That's been mentioned before. Visit us on the homepage. It's www.aggregates.eu. It's ongoing. If you're interested to cooperate with us, just call us or get in contact with us. We are happy for input anytime. And this is the end. Greetings from where I come from. This is some people from my lab, and sometimes we do some excursions for recreation, hiking in the close mountains of Tyrol. Thank you very much for your attention.
Thank you very much, Florian. That's great. We've got time for a couple of questions, I think.
Now I'm good, right? So that was very interesting work, and I found that the graph you showed very early on, where you looked at binding antibody titers in folks that acquired neutralizing antibodies versus those that didn't, and that graph predicted your prediction that in fact binding antibody appearance will be correlated heavily with the eventual appearance of neutralizing. So the question I have is, that looked like a really classic prime and boost response. I mean, that's exactly what you see as endpoint mu and gamma 3, for example, for the whole antigen, then nicely focused high affinity stuff against. So has anybody thought about looking at using deep sequencing techniques on? I recognize this is a tough study to do in people, but deep sequencing techniques to ask if the NAbs that appear subsequent to the overall BABs are clonally related and somatic mutants of some subset of those, that would be consistent with this idea that it's a prime and boost problem.
That's a good point. No, we didn't do that. So
can you get B cells from these folks, I guess is the question, and do some deep sequencing?
No, what what I think is happening is you have to have enough binding antibodies actually to have to get the selection process going later on. The patients who don't develop neutralizing antibodies always have a very low response, and if you get very sensitive with your assay, you will find binding antibodies in almost all patients. So it's some low-level, irrelevant response, perhaps pre-existing response, I don't know, against interferon-beta, and then only the ones who actually get a high titer of binding antibodies later get on with the selection process. Does that?
More or less, but it could be. I'm intrigued by the possibility that you have a priming event, and then the real problem that you face, especially given your isotype association of gamma-4 and 2b, I believe, with the new, that these are switched, probably somatically mutated things that is what you worry about. And so is the real problem a helper T cell priming event that gets these things into a germinal center and focuses them onto certain epitopes? Which should be worth knowing.
What we're looking at now in the aggregates project is look at skin biopsies early on after the first injections from interferon, see if there's a presentation in dendritic cells in the skin, early presentation. So that might be, but I can't really answer the question on the data. Bonnie?
So I have a quick question for Tim first.
I was just wondering, it's slightly related as well, obviously there's the T cell component that's just been mentioned. But do you know anything about potential SNPs in these patients and whether there's any direct relationship between genotype differences in immune responses?
No, what we looked at, not we didn't look at that. The German group looked at HLA association and neutralizing antibodies. There's some HLA types that actually make a higher risk for patients to develop antibodies, but it's very low frequency. So we're talking about 10% versus 2% of patients carrying the HLA haplotype.
So there's no obvious correlation?
No, it's no real. So far,
a foreign response as opposed to a tolerance breaking response.
Last question from Bonnie, then we'll stop that.
Am I on? Okay. So you said at the beginning you were talking about whether different types of antibodies could have different types of effects, as opposed to just the concentration of antibodies. And given that interferon has a fairly short half-life, I was wondering if there was any indication that some antibody responses can actually enhance the pharmacodynamic effect, because, perhaps an antibody binding could actually keep the interferon around for a longer period of time.
There was one very early report on this saying that there might be a prolonged PT response in patients with antibodies, but nobody else actually could find this later on. We never saw this, that there's a difference. It's always, it's gone. So if you have enough antibodies, if they go above a certain level of neutralizing antibodies, you don't find PT responses in these patients. Nothing at all. So it's really neutralizing.
Well, thank you very much, Florian. Really appreciate that. We're going to break now for coffee.